Hello, this is Gina Piscatelli with your third lecture for week 3, Monday, September 14th through Sunday, September 20th. This lecture is on membrane transport. Your reading assignment is Chapter 3, Section 3 and 4. And you have a study guide question number 16 and 18. So this lecture is all about the movement of substances across a membrane, and you can see from the cartoon that the little water molecule is saying, yeah, the aquaporin is under construction again, so I'm going to squeeze through the membrane, but I might lose you. And that's because water can't actually go through the phospholipid bilayer. So the definition of membrane transport is kind of far-reaching. It includes all the mechanisms that regulate the passage of water, ions, and small molecules through biological membranes. This includes small hydrophilic lipids and small hydrophobic lipids and small hydrophilic ions and molecules. So all of these examples show a compound entering a cell, but many transport mechanisms can occur in either direction. Notice that each compound goes across the membrane by a different route. The small hydrophobic steroids can cross the phospholipid bilayer directly. Glucose, the ion sodium, and water are all polar hydrophilic compounds or ions. Therefore, they need to go through a protein channel of some kind. Glucose goes through a glucose transporter, which is different than the protein that forms an ion channel, and is still different from the protein that forms a channel for water. The name of the channel through which water can be transported is called an aquaporin. Now there's several factors that influence membrane transport, in addition, of course, to the characteristic of the cell membrane and the polarity of the solute. Size is a big factor. Another big factor is the presence or absence of a concentration gradient across the membrane, as well as energy requirements and availability in the cell. So the first thing to consider is the solute size. If it's small, it could get across. The second thing to consider is its characteristic in terms of polarity. Is it hydrophobic or hydrophilic? And that will help determine where it goes across the membrane, through a protein or the phospholipid bilayer. Then we have to make sure that there is a protein specific for that solute. Finally, we need to consider whether or not energy is required and whether there's a concentration gradient. So before I get into all the ways that membrane transport can take place, let's first look at some terminology and some basics. Okay, so let's do concentration gradient first. Notice that in the video on the left, the substance is moving, the little red guy with eyes, is moving across the cell membrane from left to right, from an area of high concentration to an area of low concentration. So we would call that down a concentration gradient. The picture on the right, we have a substance, it's not really moving. There we go. A substance that's moving from right to left, which is kind of irrelevant, but it's moving from an area of low concentration to an area of high concentration. So this substance is moving up a concentration gradient, and that's going to require energy in the form of ATP. The first example when the substance is moving down a concentration gradient. If the substance has a way to get across the membrane, it will not require energy. So we're going to look at membrane transporters next and talk about the ways that they allow materials to pass and some of their characteristics. So first of all, every membrane transporter could be called a carrier or a channel or a pore. Whatever it's called, it's a transmembrane protein, and it's specific for a certain molecule. We say that membrane transporters exhibit specificity, and it has to do with their amino acid sequence, their primary structure. Only a certain type of molecule will pass through. That membrane transporter may or may not bind to the substance being transported. It varies, and we'll look at two types, binding and not binding. And then different cells in your body have different transporters. So not every cell in your body will transport the same substance. For example, your liver takes up glucose to store it. Your skeletal muscles prefer glucose. Your brain prefers glucose. Now, there are other transporters for glucose than just one. And so those organs are going to have multiple different types of glucose transporters, but not all of the cells in your body will. Also, the number of transporters on a cell could vary during its lifetime, and the transporter might not always be open. If it can open and close, we call it gated. So we say that the membrane transporter is gated, and it opens or closes depending on an outside factor. This picture on the bottom right shows a membrane transporter when it's open and when it's closed. When it's open, the pink substance can cross the membrane. When it's closed, the pink substance cannot. And we'll look at why it would open and close in a little bit. So this shows you the specificity of membrane transporters. Let's assume on the picture number on the left, cell number 1, that this cell has been exposed to two different kinds of molecules, the green square and the yellow circle. This particular red transmembrane protein or transporter only allows one type of substance across. Even if it's open, it doesn't matter. It cannot allow this green square molecule to go through. The same is true in cell number 2. In cell number 2, this particular transporter only allows the green square to go through, not the yellow triangle. Not that I know exactly what those are, but you know. Okay, so now some terms about what do we call these transmembrane proteins. If the substance does not physically bind to the protein, we often call it a channel. You can call it a transporter too. That's generic, but it's called a channel. If instead the substance does physically bind, then we call it a carrier. And that has to do with the type of mechanism that's going to be involved. So carriers can be saturated. Since binding has to happen, they may not allow transport as quickly as channels do. And channels could be gated or non-gated. Carriers typically are just open. So we're going to look at those examples. So here's an example of a non-gated channel, and that is the ubiquitous aquaporin, present in all of your cells, that allows water to pass easily across a cell membrane. Now it is true that water can pass through a phospholipid bilayer, but it isn't easy, and it doesn't happen everywhere in the body. It depends on the concentration gradients. So for our purposes, we're going to assume that water needs an aquaporin to get across a cell membrane. Here's an example of a non-gated carrier protein or transporter. It's always open, and a solute physically binds. So first notice you have a concentration gradient. So you have a lot of substance, the red substance, in the extracellular fluid. It will physically bind to the transporter and then be released and moved to the inside of the cell. The main point is that carrier proteins physically bind. Now I'm going to look at a channel that is gated, unlike the aquaporin. So these ion channels, there's two different ways they can be gated. This is the first way. They can be triggered to open by a chemical, and that chemical is called a ligand. I'll circle it here. That's the ligand. Therefore, we say that this channel is ligand gated. And what occurs is, this is our membrane. This purple thing is the transporter that's closed. Okay, or the channel that's closed. The only way the channel will open is if the ligand physically binds to it, and now it opens, and now a substance can move through. The example I'm showing you here is calcium, a calcium channel that will allow the movement of calcium across a membrane, but only if there is a certain substance bound. This is important in your muscle cells. So your nerves will release a ligand that binds to a muscle cell, opening a channel to let calcium in so that contraction can happen. And we'll talk about that again later. Another way that ion channels can be opened or closed or gated is through charge or voltage. So when the cell's voltage changes, a channel might open. So I need a different color here. Let's use orange. Okay, so in this picture on the left, I'm showing you where the channel is closed, because it looks open on the top, but it's closed on the bottom to the inside of the cell. And notice on the right, it's open. Okay, so that's where it's open. Now, how does that occur? It happens when the inside of the cell becomes positive. So we're going to look at the first picture on the left and notice that on the outside of the cell there is a positive charge, and on the inside of the cell there's a negative charge. This is actually true for most of your cells, actually all of your cells. But your nerves can change this. They do this by by changing the charge inside the cell. So if the inside of the cell now becomes positive, whereas it used to be negative, so now it's positive, now the channel will open. Now what's being transported is not the charge. What's being transported is a different substance, and it's sodium. So we say that this is a sodium channel, but it's gated by voltage. So it's a voltage gated sodium channel. A lot of new terms, right? So those are all the terms that you need to understand the mechanisms. And we will review the terms as we go through the mechanisms. So there are 5 different membrane transport mechanisms, maybe 6. We could count the 6th one. I don't know if we want to or not. It's a little different. 3 of the transport mechanisms do not require energy. So no ATP is needed. No ATP. These include diffusion, something called facilitated diffusion, and osmosis. So we're going to go through those first, and then I'll come back to this bottom half. So let's do diffusion first. Diffusion is defined as the movement of a solute, so a molecule or an ion, that moves across a membrane from an area of high concentration to an area of low concentration, like these sheep going from where they're crowded to where they're not. Now, there's no guarantee that sheep are actually smart enough to do that, but so to speak, they go through the membrane, which is the fence, over to where it's less concentrated. So in order for this to happen, that gate had to be open. So we know that the cell membrane has to be permeable to the solute. And you have to have this gradient in place, high to low. It's imperative. Otherwise, diffusion will not take place. So when we talk about diffusion across a membrane, it can happen in either direction, to the outside of the cell or to the inside of the cell. Either way, it all depends on the direction of the concentration gradient. And when we meet live together, we'll talk about which way the solute moves, okay? But for now, we're just gonna, okay, I'm gonna do it now. Okay, so in the picture on the left, notice that there's high concentration on the outside, low concentration on the inside. Therefore, the solute is going to move into the cell. The picture on the right, we have the opposite situation. We have high concentration on the inside, low concentration on the outside, so diffusion will happen to the outside of the cell, regardless of what the solute is, as long as it can get across. And in this example, I'm showing you that it needs a channel. It could go through here, but then it won't be charged. It'll be nonpolar, like a steroid. Now, the rate of diffusion will depend on the steepness of the concentration gradient. So, in the example on the top, this top picture, it's called a steep gradient. Notice that the red compound, whatever it is, the circle, the red circle, exists on the left side of the membrane, but not at all on the right. So this rate of diffusion will be quite fast. Okay, so it'll happen quickly. But if instead you still had more on the left than what you do on the right, 1, 2, 3, 4, 5 on the left, 3 on the right, it'll still move to the right, but it won't happen as quickly. So we call that the difference in steepness of the gradient. The steeper the gradient, or the greater the difference across the membrane, the quicker it will happen. Now, facilitated diffusion is not that different. You still have to have a concentration gradient, and the substance still moves from high to low. So a substance is going to go across a membrane from high concentration to low concentration. The difference is that you have to have, in facilitated diffusion, this is the one example where you have this carrier molecule. And this carrier molecule physically binds to the substance being transported, and so time is needed. It takes a little bit of time, and the transporters, or the carriers, I should call them carriers because that's what they are, the carrier could become saturated. Okay, so if it becomes saturated, what that means is no matter how steep, how many little red dots you put here, you aren't going to increase the rate because they're all full. This happens with a lot of pharmaceuticals. So increasing a dose won't make a difference. Other times it does make a difference, but it changes the cell permanently. Well, not permanently, but it changes that cell. So increasing the steepness of the concentration gradient doesn't increase the rate of diffusion at all. But no ATP was required because the concentration gradient goes from high to low. That's the direction the substance moves, and no energy is required. Now, the last type of passive process that doesn't require energy is osmosis. Now, the definition of osmosis is the diffusion of water. So, the diffusion of water across a membrane. And we've already decided, or I decided for you, that we were going to assume that it needed an aquaporin to do so. Okay, before I finish talking about this osmosis here, I want to talk about a joke that's said quite a bit. Okay, so have you ever heard people talk about how they're going to sleep on their books and hopefully they'll learn the material by osmosis? You know, that kind of thing. I can't think of another example. That's the main example I hear, but that's because I'm a teacher, right? That is impossible. You cannot learn by osmosis. Unless, of course, your learning involves absorbing water into your tissues. What would be a better term? I'm learning by what? Knowledge is what's being absorbed. So what would you use? Because you're not absorbing water. You would use the term diffusion. So this is everywhere in the world where they say, I mean, television, people I know, my family, my friends, probably me before I started studying this. You cannot learn by osmosis. It is impossible. Okay, but you could by diffusion, I suppose. All right, so the conditions for osmosis include that you have a semi-permeable membrane. And what that means is it is permeable to water, but not permeable to a solute. So if we look at this picture over here, the solute is a green pentagon, five-sided figure. Okay, so the solute cannot get across at all. There is no channel for it. I'm telling you this because I made it up, but this does exist, of course. So the solute cannot move. And a concentration gradient is building inside the cell where you're getting more and more production of this green stuff. Maybe it's a waste product and it can't get out. But there are aquaporins in all of your cells. And so what happens is the water will move into the region that has the higher solute concentration to change its concentration. Now, of course, that will cause an increase in volume. So if you're inside of a cell, that could cause a problem. The cell could burst. It's possible. It does happen. And we'll talk about that too. Okay, so a practical example that happens in the body in relation to osmosis. We want to talk about red blood cells. I think that would be the best. So to do this, we need to understand what osmotic pressure and what tonicity means. Osmotic pressure means the force of water. So, you know, if water builds up behind a dam, it has a lot of osmotic pressure. Tonicity refers to the solute. Okay, so if you have a lot of solute inside of a cell, that's hypertonic. If you don't have very much solute inside the cell, but you have more on the outside, then the cell is hypotonic. So we want to talk about those terms, make sure you understand what they mean. A hypertonic solution has a higher solute concentration than another solution or maybe the cell's cytoplasm. You always have to define what two regions you're talking about when you're comparing the tonicity of a solution. A hypotonic has a lower solute concentration than another solution or the cytoplasm. But there is another example, and that is an isotonic solution. Iso means same. So both solutions, or the outside of the cell and the inside of the cell, have the same concentration of a particular solute. So let's try this as an example. The picture in A, in the beaker, the solution compared to the cell inside, is it isotonic, hypertonic, or hypotonic? The solution is hyper. In B, the solution compared to the cell is hypotonic. And in C, I tried to make it isotonic with all my little dots. So there's 3 different conditions, and I want to show you the clinical relevance of that and why when someone goes to the emergency room because they're dehydrated, we don't just give them pure water, because then their cells would burst. But anyway, so if the solution inside the cell has the same concentration as outside, we would say it's isotonic. But what would happen if you submerge this red blood cell into a hypertonic solution? In that case, what would happen is that water would leave the cell. H2O would leave the cell because it's going to be sitting in a very concentrated solution. And so the cells shrivel or shrink, whatever you want to say. Isotonic is no problem, same concentration. Now, what happens if you put a red blood cell into pure water, which is hypotonic, because the inside of the cell has particles, ions, and molecules. Now water is going to go into the cell. Okay, so if water goes in, the cell will swell and burst. So that's why it's important to understand osmosis and the concentration of the solution that cells are sitting inside of. Okay, so we've talked about the passive processes: diffusion, facilitated diffusion, and osmosis. We've done that. Check, check, check. Now we're going to look at active processes. And really there's 2 that are true membrane transport processes. These will both require ATP. And what happens is the cell breaks down the ATP, which releases a lot of energy. And that energy is used to move something up its concentration gradient. So that's what we're going to look at next. Active transport processes. Substances are going to move against a concentration gradient, which means up a gradient. And of course it requires the breakdown of ATP. The 2 types are primary active transport and secondary active transport. So let's do primary first. A solute moves against its gradient. ATP has to be broken down for it to move. So let's look at the picture on the left. This is the inside of the cell where it says cytoplasm. Notice that ATP has been broken down. That releases energy, although it's not really shown in this picture. The energy is used to move sodium from low to high concentration. So no matter how much sodium is sitting out here, more can still be pumped. This is very helpful for cells that want to store something, okay, or they want to get rid of something, either way. So let's look at another example, and this occurs in your stomach. So when your stomach acid is produced, hydrogen ions are going to be pumped out of the cell into the lumen of your stomach. So this is the lumen of your stomach, and that'll become part of the stomach acid, of course. which is HCl and some other stuff. So there's a pump that does this, but in order for the pump to actually get the hydrogen to move, it needs ATP to be broken down. And then something can move against its gradient. It's like pushing something uphill. You need more energy to do it. All of your cells have a pump called the Sodium-Potassium Pump that uses primary active transport to pump both sodium and potassium simultaneously. And what it does, ATP is needed, of course. You have to break down ATP. It's required, absolutely required. What it does is it pumps 3 sodiums out continuously and 2 potassiums in, no matter what the gradient is. Always there will be much more sodium on the outside than on the inside, and a lot more potassium inside your cells than outside because of this sodium-potassium pump. All of your cells have this, okay? But what's interesting is that our nerve cells, neurons, and our muscle cells take advantage of the sodium gradient that has been established and the potassium gradient, but starting with the sodium gradient. So that gradient can be used, and that gradient will be used in our next method, which is called secondary active transport. So active transport refers to the fact that, we need some ATP to be broken down, but not for the direct movement of what's going to move. So let's talk about secondary active transport. In this case, one substance is going to move against its gradient, just like before. And in my picture here, the substance that's moving against its gradient is glucose. And I tried to make more glucoses inside than outside. So glucose is going to move through this little transporter against its gradient. Okay, but ATP is not used for that to happen directly. This little purple transporter doesn't use ATP energy. Instead, what it's doing is it is moving another substance down its gradient, and that's sodium. So sodium's going downhill from an area of high to an area of low. And that just provides enough kinetic energy for glucose to also move. So it's just kinetic energy. And the movement of sodium fuels the movement of glucose. But for sodium to diffuse down its gradient, a gradient had to be established. And so the gradient when the gradient is established is when you use ATP. So this is the whole picture. What we were looking at before was the top of the cell where glucose and sodium are moving in together, and that did not require ATP directly because sodium's going down a gradient and glucose, even though it's going up, has enough energy. It's a break-even situation. But in order for there to be a sodium gradient, we have to break down ATP, and that's done at the bottom here, and that's the sodium-potassium pump. So sodium is continually pumped out and develops a high concentration on the outside, but elsewhere in the cell it's able to get back in and it just diffuses freely. This is called a sodium glucose transporter, and it's kind of important in diabetes and just glucose balance in general in your body. Now sometimes in secondary active transport, the two substances move together. We call that a symport. So like with our sodium and our glucose, it goes together. Other times the two substances will move in opposite directions, but it works the same way. You've got one substance moving down its concentration gradient and the other substance moving against its concentration gradient. So those are all the complicated mechanisms involving ATP, or the use of energy for membrane transport. Now let's look at this last example that I also called an active process. There's no doubt that it requires energy expenditure, but it's not exactly the transport of a substance across a membrane. So it's a little different, but a cell can take something in or expel something by these methods. And it's called endocytosis or exocytosis. So let's look at those two. So first I'll define the terms. What do these things mean? Exocytosis means that the cell is expelling something. Endocytosis means the cell is taking something in. That's what endo means, within. Okay? Now, what happens is, well, it's used for bulk transport. Large materials, even a whole cell, like if it's a white blood cell engulfing a bacterial cell. Bulk transport, lots of stuff. And what happens is that for exocytosis, secretory vesicles are formed, and then they merge with the cell membrane and release. So this is just like we talked about already with proteins leaving a cell. But the cell can use this, and it does require energy to form a vesicle and all of that. Endocytosis is a method by which substances can be ingested, and what happens is the substance usually docks on the membrane invaginates and forms what we generally call an endocytotic vesicle. It doesn't really matter. So this is a form of cell eating, right? So if the substance is solid, we call it phagocytosis. And solid is a loose term because we are talking about chemical compounds here. We aren't talking about, like cottage cheese or something. But pinocytosis is usually referred to when we're talking about a cell ingesting a liquid. Okay, so I'll show you these pictures that relate to them. So endocytosis, there's three types: phagocytosis, pinocytosis, and receptor-mediated. Phagocytosis is cell eating. Notice they've got this nice big food particle here, which is a little bit anthropomorphic, but anyway, nonetheless, something solid is being ingested. In pinocytosis, this is a liquid, or at least the compound is smaller. Same process though. Receptor-mediated endocytosis is almost identical. The difference is that the substance actually physically binds to a protein receptor on the membrane first. That has significance particularly in the immune system and antibodies. So it might come in handy later, but I don't think we're going to encounter it this semester for a while. That is the end of your membrane transport lecture. Thank you very much. And there's a worksheet that goes with this that we'll talk about in class together this week. Thank you.